Hello everyone, welcome to the problem solving session 10 for the course Animal Physiology, which is an NPTEL course. And this is continuation from the previous session. So so far we have been discussing about, first we discussed about skeletal system, then we discussed about neural system. And like last time, if you remember, we did some specialized system, for example, hearing system and the olfaction system and taste and visual system, right? So now in the same theme, we will now move ahead to another such specialized system, but which is more to do with the heart, so which is the cardiovascular system, right? So if you remember in your 10th week, we discussed about cardiovascular system, and we had 5 lectures which were upon the structure and function of the heart. And then the next lecture followed the, just a second. Yeah, the next lecture followed the conduction circuit of the heart. So basically we got to see how does the conduction happen? And then we saw the contractile system. So how does the muscle contract and what are the impulses for them? And how does that get started? How does that get relayed to other, as we will see, the compartments? And then lastly, we saw what is EKG or the electrocardiogram. And we understood what is a pacemaker cell and what is a working cell. So basically, we understood the EKG and the workings of the heart. And as you would have remembered, here we are talking about contractions of different part of the heart. So we will be discussing that. And so like you can see, there are a lot of topics which are interconnected as always. So in this session, unlike the other sessions, I don't have clear partitioning on these different topics. So the topics are mixed and matched so that because it's interconnected, so it will also help you revise and see the flow between these. And I have put multiple type of questions. So for example, I have MCQs, I have true and false, match the following, fill-ups. So all so that you will find repetitive questions within these different subcategories or among these different subcategories. For example, you will see a question which appears in fill-up also, as well as in the MCQ, but that's just a different way of asking the same question. So I just wanted you to understand the subject as objectively as you can, right? Okay, but before I move on to these actual work or actual questions that I have put up, I will just quickly revise with you what you have studied in this week. The 5 lectures, I will try to summarize them and try to give you an overview of what exactly the different topics are trying to convey to you, right? Okay, let's begin. Right. So the first thing, as you can see, is we begin with the anatomy of heart, as we have been doing so far for all the system. Now, if you have to define heart, heart is a muscular organ and it is located in the thoracic cavity, and it is basically just between the lungs. So it's somewhere on the like thoracic cavity in towards your left side. And what is the function of heart? So heart functions as a double pump. Now, what is double pump? Double pump, as you can understand, it is pumping out something. What is that something? That something is blood. So heart is pumping out blood. Now, where is it pumping out to? So first is, so I hope you know that, and I can see in the figure here also that heart has 2 sides. One is this side, one is this side. So these 2 sides do different kind of pumping. So your one side sends blood to the lungs, which is because the blood is being sent to the lung. That part is called pulmonary circulation. Circulation is basically the movement of blood or the flow of blood. Now, when the heart is pumping out blood to the lungs, it's pulmonary because it's going to the lungs. So lungs is also called pulmonary circulation. And the other side of the heart sends out blood to the body. Now, because it goes to the body, it's called systemic circulation. So you have 2 types of circulation, pulmonary and systemic circulation. Okay. Now, what you don't see in this slide here clearly is that heart has 3 layers: epicardium, myocardium, endocardium. So epi, as the name suggests, it is the outermost layer. Okay, it's also called also part of the pericardium, which is basically lining the heart muscles, and it has blood vessels and fat. Myocardium, as the name suggests, it is the middle layer of the cardiac muscle and it is responsible for contraction. So we'll read about contraction. It's from these cells here. And then you have endocardium, which is the interior of the heart layer. And this is basically, as we will see in within the next few seconds, it basically lines the chambers and the walls that are part of the heart, and it is continuous with the blood vessels, right? So what are these chambers and hearts? As you can see here, say chambers and walls. So chambers are these four regions in the heart. You have right atrium, you have right ventricle. So the top portion is called atrium. The later chambers are called the ventricle. So this right is the right on your heart. So basically the image here is to be represented in the form of how it is present on your body. So basically what it means, ideally, if you are looking into this heart, this should be your right side because your right hand is this side and this should be your left. But when you draw a heart out or in the, you do the figure, you always show the right at this side. So what you should imagine is this figure of the heart is basically a reflection of what is in the present inside your body. So right atrium, right ventricle, left atrium, left ventricle. And then you have several valves, which are basically tricuspid valves, mitral valves. Let me just name out the valves first. You have aortic valve, and then you have these septums, which are basically the division, which is basically the interventricular septum. Now, what are these septums and what are these valves? So basically, so let me first tell you about right atrium, right ventricle, so that we already get started with this. So your right heart, right part of the heart, right atrium. And so everything, I know you know already, but let me just revise it for you. The receiving of the blood is from the atrium. So the atrium receives the blood, gives it to ventricle, and the ventricle passes it out to the body. So it's at atrial to ventrium, and then atrium to ventricle, and then it passes out the body. Atrium receives the blood. This much is easy, and this is what you should remember. Now, the right atrium, which is your right side of the body, is for your deoxygenated blood. The left atrium or the left side of your body, heart is basically for the oxygenated blood. So the right atrium being atrium, it receives, what does it receive? It receives deoxygenated blood. Now, where will you have deoxygenated blood? You basically, these are those blood which are rich in carbon dioxide. So you need to remove the, so you basically, it's linked to the pulmonary circulation is basically linked for your inhalation and exhalation. So when you exhale carbon dioxide. Where are those carbon dioxide taken from? They are taken from the blood vessels, right? So your carbon dioxide is exhaled out and you inhale oxygen, which is given to the blood vessels so that it can carry to the blood, different body parts. This is called purification of blood. And the deoxygenated blood is basically which is deprived of oxygen, which is more rich in carbon dioxide, right? So the right part of your body. So see, I keep saying body, right part of your heart. So you just need to remember heart is just a pumping organ. The only purpose heart is supposed to be doing is just pumping blood. Okay, and that has to be done properly. That's why all the problems that you have. But the idea of heart is to pump blood. Okay, now if it receives blood, it gives it to ventricle, ventricle throws it out. Okay, now if it receives deoxygenated blood, what should it do? It should purify this. So whom should it send? It should send to lungs above. If it receives oxygenated blood, where does it send to? It sends to the body. So it gives it out to the systemic circulation. This is how you can connect and this is how you can remember. Okay, but the most important distinction that you need to make in your mind is right is for deoxygenated and left is for oxygenated. Yeah. So right, then just to give you the idea of how the blood flows, your right atrium has deoxygenated blood, which is received from the body. Received via which? Received via the superior and the inferior vena cava. Now, what is superior vena cava and what is inferior vena cava? These are the blood vessels which will bring the deoxygenated blood. One which makes it enter from this side, one which makes it enter from this side, superior and inferior. They give it to the right atrium, right? From right atrium, it will then pass to the right ventricle. Now, who should the right ventricle give it out to? Another distinction that you need to remember so that you can not forget here. Generally, you will say artery is red in color. It's rich in oxygen. The veins which you see here as blue color, those are the ones which are deprived of oxygen. They are deoxygenated blood, right? But the distinction that you need to remember is when blood moves out of heart to the lung. Now, I just said the right atrium receives a deoxygenated blood, and from the ventricle, it has to go to the lung so that the deoxygenated part is removed. So ideally, a deoxygenated blood should be carried by vein. But only here from the right ventricle, when it is moving towards lung, it is called pulmonary artery. So the right ventricle, as you can see here, the right ventricle gives out the blood to pulmonary artery, the one in blue color. Okay. So the circle for your pulmonary circulation is vena cava. Vena cava to right atrium to right ventricle to pulmonary artery. Now the left side is for oxygenated blood, right? So your left atrium receives the blood from lungs, because lungs has already cleared, removed the carbon dioxide given the oxygen. What does it, which blood vessel brings it? The blood vessel that brings it to the atrium are the pulmonary veins, the opposite. Okay, so pulmonary veins bring it to left atrium, it goes to the left ventricle, and which is the main blood vessel that pumps to the entire body? That is aorta, right? So you need to remember it in this circulation. In the systemic circulation, the blood is brought by pulmonary vein, moves into the left atrium, to the left ventricle, and then to the aorta. Okay. All right. Now, I already spoke about valves to you. So just to give you what does valve do. Valves are basically these muscles which basically close, right? So they divide two regions, of course, and they ensure unidirectional flow. So let's say there is a flow. So there are AV valves, right? So you can see aortic valves, you can see tricuspid valves, you can see mitral valves. So here, the tricuspid valves here are called atrioventricular valves. Now, what does it mean? It means that they are between the atrium and the ventricle. What does it do? It ensures unidirectional flow. Why? Because the blood has to move from atrium to ventricle. So as soon as the blood moves from atrium to ventricle, these valves close. So what do they ensure? That the blood do not go back into the atrium. So the blood has to go out into the respected blood vessels from the ventriculum. So your AV valve basically ensures that the blood doesn't have a black backflow, right? Now, you need to remember the naming. If it is in the right side, it's called tricuspid valve. If it is in the left side, it's called mitral or bicuspid valve, right? Similarly, you also have semilunar valves. These are present in the, so you can see here, they are present here in the aortic valve. They are also present here, which is the uh, basically the pulmonary valve. So you have different valves. The only activity of valve is to prevent the backflow. So wherever they are, they ensure that the blood keeps flowing to the next target and do not come back into the previous region. Okay. So that's about it. And then we will, as we will move ahead, we will see that there are some nodes, SA nodes, AV nodes, which are responsible for your conduction of impulse. So we'll do that. Let me just move ahead from the anatomy, right? So anatomy, and during anatomy, I've already explained you how does the blood flow. So the numbers on this side of your figures basically tell you how does the blood flow via the heart. And because I am talking about heart and your blood flow together, it's called cardiovascular system. Now, as the 1, 2, 3, 4, 5, 6, and 7, the color of how to read this figure is basically how do you see the color of the arrow? If it's red, it's for oxygenated blood. If it's blue, it's for your deoxygenated blood. If it's blue, it's on, and it has to be on your right. If it's red, it has to be on your left, right? So it's right, it is left. Please don't forget that. That's very important. And basically, let me just move over via one of these flow. So let's say about this. This is the deoxygenated blood. So it is coming from the body. So via vena cava, superior vena cava, it goes into the right. So basically, it comes via pulmonary artery. It goes via, I'm sorry, hold on. Wait a minute. Right. So the first one is for to the right heart. via the superior vena cava, which is basically bringing from the all the different body parts, enters atrium, enters ventricle, from there goes to the pulmonary arteries and it moves out. That's one circulation. Now, from your pulmonary veins, you have the oxygenated blood coming into left atrium, goes to left ventricle, and then moves to or leaves the heart via aorta, right? So again, you have this aorta, you have moving out aorta, and then it moves out to the different part of your body. Now, aorta just can't be at the bottom, because if it's bottom, it will only take to the trunks and lumps. So you have the aorta here as well. So this is aorta, part of aorta, and the other part of aorta. This goes to heads and arms. This goes to trunk and limb. Similarly, to your vena cava, you have superior vena cava and you have inferior vena cava to collect the blood from all the different body part. Right. So that's the whole idea there. Okay. Now moving ahead. So these different contractions or different movement of blood has distinct name. Okay. So first, and this is something that you would have seen in your blood pressure, 120 by 80, or when we say, we don't see 120 by 80, we see something like 150 by 90, we say the person is having high blood pressure, or we see substantially lower than 120 by 80, we say low blood pressure. So the blood pressure are basically the systolic and diastolic pressure that we will talk about now. So what is diastole? Just a second. Yeah. So what is diastole? Diastole is basically all chambers are relaxed and blood flows into the heart. Now, what is relaxed in the what terms? Basically, when your heart is pumping blood, it is contracting. When your heart is not pumping blood, when the uh, muscles or the cardiac muscles are not moving, they're relaxing. They're basically not yet started to contract and send the heart blood out. So what does the heart want is, as soon as the blood enters any of the particular chamber, the heart muscles or the heart chamber is filled up, and that kind of builds up pressure, and the walls open up and the blood flows down, right? So until this filling is done, it is relaxing, right? So that's what, when it is diastole, your all chambers are relaxed and the blood is flowing into the heart. It is coming, right? Then you have atrial systole, but ventricular diastole, which is basically the atria will now contract, pushing the blood into the ventricle. The first thing is atria has to put it to the ventricles. So the atrials will now contract, and that's called systole, right? Following atrial systole, you have atrial diastole and ventricular systole, which is basically when the atria has pumped out the blood, it will now relax. Now the ventricles have to contract because it has filled up with blood, and it will push the blood out. Right, and then both of them will come back to cardiac diastole. So that is your diastole and that is your systole. Now I will give you just 30 seconds to imagine 120, if you know which one is systole and which one is diastole when I talk about 120 by 180. Right? So 120 by 120 by 80, I'm sorry, 120 by 80 is basically the, why am I keeping saying these normal, this 120 by 80, because 120 by 80 is your normal blood pressure, right? Here, the upper value, which is the 120, is for the systoles, contractions, and the lower, which is 80, is basically for the diastoles. Okay, right. Now, moving ahead. Now, whatever I just told you is shown in this schematic here. So let me just walk you through. And this timing here shows how quickly these contractions are taking place, right? So this entire cycle of diastole systole is basically starting with atrial systole, or you can say it starts with the diastole, but the actual process of pumping blood starts from, or contraction starts from atrial systole. So your atrial systole is basically atrial contraction with which forces a small amount of blood, additional blood into relaxed ventricles. That follows by ending of systole and the beginning of atrial diastole. Understandable, it is pumping out blood and it is relaxing. Next is ventricular systole, first phase. Now, what is first phase? So your ventricular contraction, any systole is contraction. So it pushes AV walls closed, but does not create enough pressure to open semilunar walls. Now, what does it say? It says that when your blood has flowed to maintain the unidirectionality, the first contraction is for these valves to close back. And then the blood will spread up, and then it will create enough pressure for the semilunar walls to open and these blood to move into the blood vessels, right? That's what it means. So that's why first phase and second phase. Second phase is as ventricular pressure rises, excess pressures in the arteries, the semilunar valves open up and the blood is ejected. As soon as that is ejected, your ventricular diastole begins. And then basically when it relaxes, both the atrial and ventricles are relaxing. That's called diastole, cardiac diastole. Okay, now we come to cardiac conduction system. Now this is important because here we will talk about how are these impulses taking place, how are these conduction taking place, right? So what is this cardiac conduction system if you have to explain? So the cardiac conduction system is basically a specialized network of cells which generate and conduct electric impulse. Now, again, like I have told you in the previous sessions also, the idea of electric impulse is the same sodium depolarization, influx, sodium depolarization, potassium efflux, depolarization, repolarization. We have understood this concept at the beginning for this very reason, because once you understand the impulse, it's very easy to apply in all the other system because all the system have this underlying action potential that generates all the action potential and lets all these activities take place. Okay, right. So in heart also you have the rhythmic contraction of heart taking place because of this electric impulse. And what does rhythmic contraction in other word mean? It means proper pumping of blood, right? Okay, so in this regard, there are some terms that you should remember. For example, SA node or sinoatrial node, we will do that. Then there's AV node or the atrioventricular node. Then you have bundle of His. Then you have bundle branches. And then you have Purkinje fibers. Okay, now let me tell you what does each of these do. Where, first of all, where are they located? So the SA node is here. Sinoatrial node or the SA node is basically here. Where is this? This is basically in the right atrium, right atrial wall. Okay, near the superior vena cava. So where your superior vena cava is attached, it's right present at right that place. And it's called pacemaker of the cell. Now, what is pacemaker? The name also suggests, right? It's basically that maintains a rhythmic contraction of blood. It basically generates those impulses. And because it generates impulse, it's called the pacemakers of the heart cell. Okay. Next, you have the AV node, which is present in the interarterial septum near tricuspid valve. So where is your AV node? Your AV node is here. Okay. So this is your AV node. Okay. So this AV node is basically, as we will see, it is present in the interarterial. So as the name suggests, interatrial septum. So it's here, right? Okay, and near the tricuspid valve, which is present here, as we know. This one is tricuspid valve. This one is bicuspid valve, or also called the mitral valve. Okay. So AV node. What is AV node? Atrioventricular node. The name suggests what it's supposed to do. The next node is bundle of His. This is present in the interventricular septum. So one node is here, one node is here. Intraventricular septum. Your bundle of His is here. Okay. And then you have these left and right branches. So you can see this is bundle, this is branching out on the left, branching out on the right. Okay. And then, so what are these three doing? So it's basically they are, first of all, the AV node is there for delaying the impulse to basically for facilitating the atrial contraction. So what the idea is here, you have a generation impulse. It can't be that quick that your ventricles start contracting before the atrial contraction happens completely. I hope that made sense. What I mean is, until the atria has completely cleared itself of the blood or has completely, ejected all the blood, the ventricles cannot contract. It will not anyway. That has been ensured by the closure of valves, but the valve closure should also not happen before the atrium has completely pushed it out, right? So how do you generate or how do you maintain this at the AV node? So it starts from SA node. the atrium is undergoing this this depolarization, starts contracting, and then of course, because it's electric impulse, it will come to AV node. At AV node, if it generates the next impulse or it propagates the impulse very quickly, the ventricle muscles will start contracting. So what it does is it delays, it stops the relaying of the impulse. What happens? Because it has slowed down, this contraction, which was because of the SA node, will still continue and will pump out the exact blood, and then this will allow the next set of muscles which are in the ventricles to air contract, right? So the delaying of the conduction is very important for you to remember. Okay. Now, next you have bundle of his and the bundle branches, which are basically transmitting the impulse from the AV node to the ventricles. So the ventricles, because atrium is done by SA node, from atrium till the ventricular, like contraction, you have the AV node, and then continuing in the ventricles, you have bundle of his. And then you have lastly the Purkinje fibers, which are in the ventricles walls, which are basically for putting them out into the respective blood vessels, right? So I hope that has given you an idea of how does the conduction take place. So in case it is not yet, you are not able to visualize it yet, you can see these 5 points are basically telling the same thing. So they are saying that the first step of this conduction is the impulse initiation. Of course, your SA node is where your depolarization takes place, and we know what is depolarization, basically the influx of sodium, right? And it becomes your atrial contraction. Next, you have AV node, but it is a delay at AV node, which is to ensure that your ventricles are filled with blood. Next, you have impulse propagation. I told you it will delay and then it will propagate. That is because of bundle of his. So it will pass on to bundle of his and then they will propagate it further, right? So from the bundle of his, it goes to the branches and then to the Purkinje fibers, right? And then you have the ventricular contraction, which is basically contracting out in the blood vessels. And then it has to come back to the normal state, which is via repolarization when ventricles are relaxing, right? I hope that makes sense. Right. There are some special features about pacemaker cells that you need to remember, but I will discuss them when we do the questions, right? And the last thing is this graph here, which I am sure everyone would have seen or at least heard about. It is called the ECG, which is basically monitoring the electrical activity of your heart. And you would often see these kind of graphs there. So which is called the PQRS waves, right? And then you have the distances of PR, ST, SQT, all of this. So let's try to understand what does the ECG basically do? What does it basically tell you about, right? So like I said, ECG is basically mapping the electrical activity of your heart over the time. And if you have seen when your ECG is taken, you have these different electrodes which are placed on your heart, and there is a machine which basically reads the conductivity of these electrodes, right? And what is basically all these graphs that you are seeing? These are basically the depolarization and the repolarization of atria and ventricles, right? So let me try to explain this with all these different waves. So first is this P wave, okay? Now, what is this P wave doing? It is showing atrial depolarization. Makes sense because this is the first thing that you should anyways. That basically is starting the entire procedure that we are reading, right? So your depolarization, the timing, the shape of the graph or the wave tells you about how well your SA node is functioning. Because if your SA node is firing impulse, then only your atrial contraction will take place, right? Now, it will contract, come back to its base, right? Then you have these QRS, right? So the distance between this PR interval, which is the next offshoot, this gap is basically the gap that is required or that is taken from the atrial depolarization to ventricular depolarization. Remember, we told AV node delays this depolarization. So this PR gap is delayed interval, right? And then you have this entire QRS complex. This is ventricular depolarization. The first wave was atrial depolarization. The next part is ventricular depolarization, where your impulse is now spreading from into the ventricles and the ventricles are contracting. Now, then again, when it has come back, you have this ST interval, right? Now, what does this ST interval saying? This ST interval is basically the ventricular repolarization. Makes sense. Also known as plateau phase, in the ventricular action potential. Okay. And then you have the T wave, which you can see here, right? This is when things are going back to its normalcy, which is the ventricular repolarization. So ST is the beginning of the repolarization. T wave shows the actual repolarization, right? The ventricles are relaxing now. And at last, your Q and T interval, which is basically this entire part together, is basically the total time for ventricular depolarization and repolarization. So because here your depolarization comes from the AV node to the bundle of His, and this is when it depolarizes, repolarization after depolarization. This has already done that by the time it comes here, right? So that's why this you have a PR interval, and then you have a QT completely for your ventricular polarization, repolarization, and depolarization. So that's the QT, that's the PR, that's the ST, P, QRS, and T. All of this we have discussed, right? Now, one thing which is not part of this figure here or the photos that I have just discussed, but something that will keep cropping up in a different kind of questions that you will attempt for exam and question paper or for your assignment is basically phases of these action potential and ECG correlation, right? So let me just put you through this again in terms of phases now. Okay, so let's begin with phase 0. What is phase 0? Now, obviously, you would think that this is the exact way how phases will also be going. So you feel basically this is phase 1 or phase 0, 1, 2, 3, 4. So let me just tell you how it actually goes, right? So your phase 1 is basically Na plus K, channel inactivation and K plus efflex, right? But before phase 1, you need to understand phase 0 so that it makes sense to you, right? So your phase 0 is basically the influx of sodium ions, right? Now we are only, how do you say? We are only looking into the ventricular contraction here. So phase 0 in relation to this is, of course, because we are talking about depolarization, we are talking about any influx. We are talking about the, sorry, just a second. Yeah, I am talking about the beginning of the QRS. Okay. Then you have phase 1, which is when your sodium channels inactivate and your K plus efflex happens, right? So this is your QRS. Phase 2 is there for the plateau. Phase 3 is your, so what is the property of the plateau? You have calcium ion influx, of course, because they are contracting, and your potassium ions efflux to balance out whatever has happened because of the influx of sodium, right? And then phase 3 is when your efflux is at its highest, potassium is moved out at its highest. And phase 4 is your resting potential, which is your normalcy, right? So basically, how do you see your phases in this map? Your phase 0 is basically your QRS upstroke beginning. Okay. Phase 1 is your early QRS. Phase 2 is your S and T segment. Phase 3 is your T. And phase 4 is the baseline between this T and the next P. Okay. So we are considering from here to here. And what was this? This is about ventricular repolarization and depolarization. Yeah. All right. So that's about it. Now let us begin with the questions so that we are on time and we can see as many as questions as possible. Okay. Right. So like last time, I will read out the questions, wait for 10 seconds and show you the answers. All right. Okay. Just a second. Right. Okay. So first set of slides are relatively easy and simple for you just to map it out on your imagination. So you have, of course, on column A, some terms, and on column B, the respective answers for that. So which one do you think would be the correct answers? Now, since I can only give you 10 seconds for these, what I will do is I will read out the answers. But you, when you are trying to solve it, when you are looking into the video, you can pause the video, answer yourself, and then restart the video, and you can check for yourself if you were correct or not. Right? Let me just read, or maybe give you the answers and then show you the answers correctly. Right? So your essay note is the one that spontaneously depolarizes. Now, why this spontaneous, I will tell you. I have questions on them and then I'll come back and explain you. The spontaneous depolarization of your, depolarization is required to generate the impulse. But what is spontaneous and why it is important, we will do in the coming slides. Right? But for now, SA node is the one that is responsible for spontaneous depolarization, which is also called the heart pacemaker. So the pacemaker should have given you that it is SA node. Next, the Purkinje fibers. These are basically the one that rapidly conducts to ventricular myocardium for its contraction. AV node is basically the one that delays impulse to allow atrial contraction. QRS complex is for your ventricular depolarization on EC. This makes sense, right? T at the end, so ventricular repolarization. Plateau. So plateau is when there is sustained calcium influx. Why? Because the ventricles have to contract. And the last one, which is Purkinje current, this is for sodium and calcium leak currents causing spontaneous depolarization. So it kind of gives you an idea of what I'm going to talk about, but let's hold on to the idea, right? Okay, this is easy. And I thought before I explain all of these, let me just ask you people if you can remember. So these are the type of blood vessels that you have. You have more also, but that I will come to later. But for the time being, you have 4 blood vessels: arteries, veins, capillaries, and arterioles. What are their features? Right. So your arteries are, so remember, why is this question important? Because this is talking about the individual property of each blood vessels, right? So it is talking about artery. It's talking about veins. It is talking about the pressure on each of the blood vessels, what these blood vessels are facing and why are they facing, right? So here, arteries are basically the ones which have thick muscular walls because they are supposed to carry the blood away from heart. So they have to carry it to different region. So they need thick muscular walls. Veins are the ones which have thin walls and they have valves present. So they carry, of course, from carry the blood to heart. Capillaries, as you will see in respiration system, they are the fine walled arteries which are responsible for, or fine walled blood vessels which are responsible for exchange of gases and nutrients. And the arterioles are the smallest branch of arteries that can regulate blood flow, because arteries, if you keep imagining these thick layered, if they keep going, they have to really spread out. So for spreading out, they have these small branches which are called the arterioles. Okay. All right, the next ones. So these are the 4 regions of heart that we have already discussed. So it's basically a recap for you. Right. So we already know left atrium. How can you, like the cue for you to do it very quickly is just remember which one is oxygenated. So which one is oxygenated? Your left is for oxygenated and your right. I am showing the opposite way because that's how your figures are generally, right? So your right is for deoxygenated and your left is for oxygenated. Find out the deoxygenated one, A and C. Then you need to remember receives is atrium and pumps is ventricle, right? So that's exactly what you should do. Receives deoxygenated should be your 2, right? So 2C. Pumps is next. So, sorry. Yeah. Receives is your 2C and your pumps deoxygenated blood is your 4. Similarly, receives the oxygenated blood is 1B and pumps the oxygenated blood is your left ventricle. Yeah. Okay. Now, these are some fill-ups. So I will read out the question and then wait for 10 seconds and then give you the answer. Okay. The question say the right atrium receives dash from the superior and the inferior vena cava. What kind of blood? The valve between the left atrium and the left ventricle is called what? So which valve is present there? Pacemaker of the heart is which cell or which node? Phase of the ventricular action potential. That's why we are only talking about the QT segment. So the phase that is characterized by prolonged plateau is maintained due to what? So we know there's a plateau. Why is a plateau there? And the last one is the bundle of specialized fibers that transmit cardiac impulse from the AV node to the ventricle is called what? So bundles should give you away what is the name. The name is pretty simple and easy for you to remember. Right. So coming to the answers, right, I said the right is for deoxygenated blood. The left valves or the left atrioventricular valve is mitral or bicuspid valve. Pacemaker is the SA node. The plateau phase is due to calcium influx, which is through L-type channels, and the bundles are called the bundle of His. And now you have true and false. So I will read out the 5 statements, and you need to, in your like the question asks you which one is true and which one is false. So the left ventricle pumps blood into the pulmonary artery. Purkinje fibers are located in the walls of ventricles and help propagate the action potential rapidly. The AV node initiates the heartbeat. The P wave in the ECG represents atrial depolarization. Arteries carry blood away from the heart regardless of the oxygen content. Yeah. So the answers are these. Right. So first one says the left ventricle pumps blood into pulmonary artery, which is false, right? Because the left ventricle pumps blood into the aorta. Okay. Right. So aorta is an artery. Okay. Now the next one is true. Purkinje fibers are in the last segment, so they are for the ventricles. They're propagating the action potential in ventricle. The AV node initiates the heartbeat. No, we know which one does. This is done by SA node. P wave in the ECG is for atrial depolarization. That's correct because that's what it meant to be, because it starts with the atrial depolarization. And the last one says arteries carry blood away from the heart, regardless of the oxygen content. Let me take you back to this figure here. Yes. So where is pulmonary artery taking blood to? It is taking blood to lungs from heart. Right? And what is pulmonary vein doing? It is bringing blood to the heart, right? What are otherwise blood arteries doing? This is artery. Aortas are artery. They are taking blood away from the heart. What are veins doing otherwise? They are bringing blood into the heart. So that's why when the blood reverses its direction to the heart, so to the lung, the naming switch. But the idea why they are artery and why they are vein is because of the directionality of blood. And that's why I wanted you to come to this question and then I would have explained to you. So your arteries, they carry the blood out of heart and regardless of the oxygen content. So they will have either oxygenated blood or deoxygenated blood. But because they're carrying heart blood away from the heart, they will be called arteries. So pulmonary artery, they will carry blood from the heart to the lungs for purification, but they would be deoxygenated blood. Yeah. Okay. Right. Okay. Now let's begin with these questions, which is not very difficult. So at each slide, I will read out the question, wait for 5-10 seconds, and then I'll give you the answers. Okay. The first question is the Frank-Starling law of the heart states that, right? So it states that, so this law states that the stroke volume will increase with greater venous returns. And I don't think so. This is complicated to understand. The more the blood input is there, the more the blood pumping will be there. It's as simple as that. Okay. Next, the human circulatory system is a closed loop because this was one of the first topic that professor had taught you during this week. So just touching on that topic, why do you think human circulatory system is called closed loop? That's because the blood flows inside the vessels only, right? There is no body fluid or there is no other tissue that is taking up the blood directly. So blood has to move through vessels and it remains within the vessels the entire time it is in, if it is any organ, if it is in any tissues, it stays inside the vessel. Okay. Now, which phase of your cardiac cycle corresponds to ventricular ejection? Basically, ventricular contraction basically is systole, because diastole, they are relaxing. These are easy questions. That's why I am not spending much time in waiting for the answer. Next one. Pacemaker cells maintain their resting membrane potential at what millimoles? So the answer is minus 40 millivolt. Now there is a special point here. So as you would have noticed so far that whatever neurons we have been studying, we have seen that the neurons or the myocytes generally have a minus 70, minus 80 millivolts as the resting membrane potential. But your pacemaker cells, which are your SA nodes or AV nodes, these have minus 40. However, this is not a stable resting membrane potential. What is a stable resting membrane potential? Which stays like this only until your Na plus influx begins, right? So it stays at minus 40. However, in your pacemaker cells, what happens is they have a maximum diastolic potential, which is basically when they are in diastole, it's there at minus 60 to minus 70 millivolts, right? And then they slowly depolarize till minus 40, right? And when they reach minus 40, they begin the depolarization. Now this depolarization, this further depolarization from the minus 60 to minus 40 is because of the calcium, because of the leaky current that we will see in a while. It's because of those kind of features that it can be at minus 60 and come to minus 40 and then start depolarization. So it doesn't need any other impulse to come and say, hey, you need to pump. It can on itself start pumping. Okay. All right. Right. So electrical activity of conduction circuit starts from AC node. We know that. Okay. Now calcium is important for any muscle contraction, right? So that is also regulated in cardiomyocytes from, or that is being secreted from, as we know, sarcoplasmic reticulum. Now carbon dioxide oxygen exchange at the body happens at which scale? It happens at capillary scale. Okay, the other doesn't even make sense. Now, which of these cells are found in the heart? Endothelial, cardiomyocytes, and cardiac pacemaker cells. So, which one of this? Of course, two of them bear the name cardiac. So, what about the other, endothelial? So, the answer is all of them, right? So this endothelial, they are present in the inner surface of the heart and the blood vessel, which is endocardium. So endo. Cardiomyocytes, they are basically the muscle cells. They are needed for the pumping action. And the pacemaker cells, we already know, they are the one that conduct the electrical impulses. Okay. Now, which of these has higher diameter? Which is basically, they are having higher sizes. This becomes easy if you can just imagine the picture I showed you, or the diagram that I showed you. Right. So the answer will be vena cava. And if you see the image that I was discussing on for the entire conduction, you will see the vena cava has highest diameter. It is the largest blood vessel also. It has 3 centimeter around your diameter. Aorta comes next with 2.5 diameter. Aorta is the largest artery. Okay. And the capillaries, they are the smallest. They have to be, I told you, they are for the exchange. So for exchange, you need very thin layers, right? So there are some micrometers. And pulmonary artery is again artery. So it's around 2 2 centimeters something. So again, just to recapitulate, capillaries are the smallest, they have to be. Vena cava is the largest blood vessel. Aorta is the largest artery. Pulmonary artery is like a normal artery, which is around 2 centimeter. Okay, all right. Okay, now which of these blood vessels have valves? So the answer here will be veins. Okay, so your veins have, if you see, they will have valves. Okay, and these is basically to prevent backflow of the blood against gravity. Okay, arteries, they will have thick wall, they have high pressurized walls, but they don't have valves. Okay, okay, but remember, arteries take blood out of heart. So two arteries, you have exception. One is aortic or aorta and one is pulmonary artery, right? The valves that are maintaining the directionality or unidirectionality of blood in these two blood vessels are these semilunar valves. Remember, we talked about the semilunar valves. So these valves are associated with arteries, but in general, the arteries that you see are not having any valves, but veins do have valves. Okay. Like the other two, alveolus and bronchioles, they are in your respiratory system and they don't have any valves. Okay. Okay. Next one. Now you have two successive P waves. So what does it say? So try to remember what does P wave tell you, and then try to look into the answer. So the answer is left atrial enlargement. Okay. Now what is atrial enlargement? Is basically when your left atrium, it becomes abnormally enlarged. Remember, your P wave is left atrial depolarization. And what you are seeing is you have two successive P waves. So it basically tells you that your left atrial has undergone some anomaly. It's generally in the form of your enlargement. And why do this enlargement generally take place is because of some heart associated problem, for example, blood pressure, high blood pressure, or you have some obstruction to the vent. So why is it happening two times? Because it's not properly pumping at the same time, right? So some kind of obstruction, you have too much of blood flowing into the heart. So these kind of problems leads to the enlargement of, so enlargement is basically it's increasing in size, right? And because it is increasing in size and it is not following the timing properly, so you have this impairing ventricular feeling and your atrial size increases such that you have these two successive waves. So it has done depolarization and pumping. Again, it will depolarize and pump. So something like this. So it's generally a problem with the left atrial. It's at the beginning. Yeah. Okay. Now this problem. So your P wave is, we have already seen, right? We already discussed. So one problem is two waves. The other problem is about the amplitude. Amplitude is basically the length of these tip to the base, right, of any wave. So if your P wave has decreased amplitude, so let's say it's supposed to be like this, but as we know, it's supposed to be like this. But let's see you see something like this, or see a flattened line. In this case, with a decreased amplitude, what will you understand? Hmm. Right. So the answer is hyperkalemia. Okay. Now, what does it mean? So your decreased amplitude is because of potassium imbalance. Okay. And in hyperkalemia, your P wave reduces. And along with this, you have your PR interval, which is increasing. Remember, everything is subsequent to P. And your T wave, which is at the end, also becomes very tall and peaked, right? If you see the reversed, which is basically flattened T waves, P wave is still the same, but T waves is decreasing. See, P and T are at the end, right? So if P shows problem, it's hyperkalemia. If T shows problem, it is hypokalemia, right? Now, what is exactly happening? Why kalemia and why all of these suddenly? So hyperkalemia is basically uh, your there is a how do I say, there is a problem associated with your potassium efflux. Okay, so you can have kidney issues, you can have some other kind of problems, you can have acidosis, something which is not getting your potassium ion back in the original amount, right? It is not putting it out correctly. So what happens is your such cells, the resting potential becomes less negative, right? And there is increased excitability. But at severe hyperkalemia, you also have Na plus K inactivation. So everything kind of gets very complicated. You have a lot of potassium, even though your potassium are supposed to go out. Yeah. The opposite, where your potassium is not even, like it's continuously moving out, then you have your hypokalemia, right? And it's at the end. So it's the opposite. More of potassium ions, hyperkalemia, less of potassium ions, hypokalemia. Okay, next question. This we have already done in the last class. So ryanodine receptor and DHPR receptors are basically calcium channels. We have already discussed that. Okay. Now, which chamber of the heart pumps oxygenated blood to the body? Remember oxygenation and deoxygenated, which part refers to what? And then try to remember. Pumps, not receives, and pumps through the body. Left ventricle. Which blood vessel carries the deoxygenated blood from the body into the right atrium? Which, in other words, it means it receives deoxygenated blood. So which one takes it to the right atrium? Vena cava, right? From the body into the right atrium. Which valve prevents backflow of blood from the left ventricle into left atrium? So basically from ventricle into atrium, but left. Mitral or bicuspid valve. Yeah. Okay. What is the main function of pulmonary arteries? Arteries, you need to remember, and pulmonary tells you the location. Carry deoxygenated blood to the lungs. Pulmonary, lungs. Artery takes blood away from the heart to the lungs. Why would it do? For the purification of blood. So that's for the deoxygenated blood. That's how if you keep it in your mind as a story, as a flow chart, it becomes easy for you to answer these questions. Okay. Next. Which side of the heart has higher pressure? What do you think? Which of the side or which of the blood vessels would have higher blood pressure? So which one has higher blood pressure? First of all, the left ventricle. Now why? So remember it like this. The left ventricle, what is it doing? It is pumping out your blood into aorta for systemic circulation, right? Now, because it is pushing out to aorta, which has to further give it out to the entire body, this requires much higher pressure to deliver the blood, right? Now compare this to right ventricle. So what does the right ventricle do? It has only to give blood to the lungs, right? So it has to just give out the blood to the lungs. So it's a very low pressure circuit. Whereas the atria, they just receive the blood and give it out to the next. So comparatively, it is very, very low pressure to work. But think about the ventricle, left ventricle, which is gives out to the systemic circulation. It has very high pressure, right? So overall in the four chambers, the highest pressure would be on the left ventricle and the lowest pressure would be on the right atrium, because right atrium is receiving blood from the vena cava, right? It is dumping. Everything is coming and dumping. It has the least pressure to maintain, right? Okay, right. Okay, now let's move to the next question. Yeah. Which blood vessel carries oxygenated blood from the lungs to the heart? Wherever you have the word lungs, One thing is obvious to you, and the other part says directionality of the heart to the heart or from the heart. That tells you the second part of the answer. So which one do you think is the answer? Pulmonary artery, pulmonary vein, aorta, or superior vena cava? Sorry, I'm taking a pause because my internet is lagging. Just a segment. Yeah, I am. Just give me 10 seconds. There's some internet issues, but yeah, in the meanwhile, you can just try to imagine where and what is the directionality of the flow of current, sorry, flow of blood in the heart, and which one would be corresponding to this. Right. I hope it works. Yeah. So pulmonary vein, right? I already discussed this with you. Sorry. Coming back. Yeah. Which blood vessel has the thickest wall to withstand high pressure? I showed you as a MCQ. Sorry, fill in the sorry, match the following. But what do you think is the answer here, and why do you think the answer is there? In the sense, which one is the correct answer, and why does it have high pressure? Right. So arteries, right? So your arteries have the thickest wall, and that's what is sustaining them to have high pressure. Now, why is that? I already discussed in terms of the left ventricle, right? So your arteries are basically carrying blood away from the heart to the entire body. So these are carrying away from the heart. So that's why they are under high pressure, right? And for that, they have their adaptations. So they have these thick muscular walls, elastic walls, which can, sustain fill up of the blood, right? So that they can pressure, they can regularize them with the pressure. While the veins, they have thinner walls, they have thinner walls and valves also, so they maintain a low pressure system. Your capillaries have to be the smallest, thinnest, so they are single layer because they have to allow exchange of gases. And you have these venules, which are vessels, right? Smaller, thin walled. These are also thin walled, but they collect the blood from the capillaries. Okay, so they're basically taking it out. Okay, now basically what I wanted you to imagine when I say why do they have blood pressure is to understand why is the artery under such high pressure. uh, just remember that this has to basically deliver the blood to the entire body, so the systemic circulation is driven by the arteries, and because they have such a high blood to like transport, they are always under high pressure. Okay, right. Okay, this already we have discussed, so this shouldn't be very difficult. Which structure prevents backflow of blood inside veins? That's valves, right? Okay, so the SA node is also called the natural pacemaker, right? Now, if you have to define natural pacemaker, it basically is talking about the SA node, which is located in the right atrium, and it is pacemaker because it is generating spontaneous electrical impulses. Why pacemaker? Because it sets the rhythm of the heartbeat. Your heartbeats that we keep hearing about the, uh, like we keep saying your heartbeat is there, you are beating 72 beats per minute. It basically these electrical impulses, right? And how do the stethoscope take this up? You will hear the lub and the lubbed up sound. That's because of the closure of the valves. Those are the sounds. But what are the sounds come are basically from the impulses, and that states the rhythmicity, right? So that is the rhythm of your heartbeat. So because it is setting the pace, that's why it's called the pacemaker, right? Okay. Next, so the AV node delays the electrical impulse. Why does it do so? We've already discussed this, but there are some options for you to understand why we do so. Why does it do so? Which is to allow the atria to contract completely before ventricle starts contracting. Okay, that makes sense, right? Now, Purkinje fibers, where are they located? Like I said, some questions will be repetitive, seems like repetitive in the fill in the blanks and in the match with the following. That's just to put you in different, like practice of answering the question. Right. So the answer here will be ventricular walls, right? Okay. So the next question is, so we know it's in the walls. What does it do? What is the function of these Purkinje fibers? So they're basically for the simultaneous contraction of both the ventricles. Remember, these are the ventricles, and so they are ensuring that your ventricles are contracting together. Okay, the bundle of His, where is it present and what does it connect? They connect the AV node to the Purkinje fiber. They're in between these, right? Okay. Coming back to the polarization and the depolarization, which ion is primarily depolarization's cause, or which is responsible for your depolarization in cardiac action potential? We know this already. It's always sodium ions for this case, for the most of the case. Okay, this is a very interesting question. So this tells you which is the correct pathway of electrical conduction in the heart. So remember what we discussed. Where does it begin and where does it go next to? Right. So we have already discussed it that your electrical impulse begins at the SA node. So it initiates the impulse. From the SA node, the impulse spreads through the atria and then reaches the AV node. Right? AV node delays it, further sends the impulse to bundle of His, right? Which is also called the atrioventricular bundle. From bundle of His via the bundle branches, it reaches or the impulse reaches the Purkinje fibers, which are responsible for the coordinated ventricular contraction. Okay? Yeah. Okay. The rapid depolarization phase of the cardiac myocyte action potential is caused by which phase? What phase? Anytime it's depolarization, you already know. But also remember, try to link here only. We already did about phase 0, right? So we are talking about depolarization, but what depolarization is phase 0 referring to? So the answer is sodium influx, yes. But can you remember which phase of your conduction system or cycle is this phase 0 referring to? Or probably the ECG graph? The beginning of the QRS complex, right? The beginning or the ventricular depolarization, right? Yeah. Similarly, So repolarization, which is phase 3 in cardiac muscle fibers, they occur due to what? And which one is phase 3? Can you remember that? Which wave, which phase, and what is the mechanism there? So the answer is potassium efflux, right? You are basically repolarization means your efflux of potassium. Which wave in the ECG shows this? The T wave, right? So this is how among different questions you can link them up so that you can, like concretely build these topics connections. Okay. The plateau phase of cardiac potential is due to what reason? So plateau phase, try to remember where is plateau phase first of all, and then try to understand. We did this question in your, I think, Phillips also. So can you remember? And where do you see in the wave the plateau phase? Right. So the answer is delayed potassium efflux balanced by calcium influx. Okay. Now, what is it saying is that it is a plateau phase. Where did we see plateau phase? We saw it in the ST segment. What was happening here is the early ventricular repolarization. So it is a repolarization initial phases. So you have the potassium efflux, which is delayed, but you have the calcium influx, which is going to push it forward in the next phase only. Yeah. Okay. So we know that you have plateau phase, calcium influx. How else do you explain this prolonged plateau phase out of these given options? Sustained calcium influx due to L-type channels, and that is delayed K plus efflux. So there is a delay and there is a sustained influx, which is together leading your. So your plateau phase is basically a repolarization phase, right? It has to begin the repolarization. Plateau means it is keeping it delayed because as soon as the repolarization happens, you have the T wave. So before T wave, you have this flat line, which is basically the gap before your repolarization starts. Yeah. Okay. Now, here is where I will discuss what I was trying to tell you before. But before that, you answer me. Pacemaker cells differ from ventricular myocytes. Why? So the answer is they lack a stable resting membrane potential. Okay, what does it mean? The first question is the pacemaker cells and the myocytes. Pacemaker cells are the ones that begin the impulses, right? Ventricular myocytes are the ones that contract. What is the difference between them? One of them have, sorry, one of them have unstable or lack a stable resting membrane potential. So let's first discuss about the pacemaker cells. Okay, so these pacemaker cells, like I have been telling you, they are able to spontaneously depolarize, right? And that's because of the unique properties. So what is that makes them different? So first of all, let's just understand in terms of millivolts also, right? So as I told you, pacemaker cells, if you remember from the first part, pacemaker cells have a less negative maximum diastolic pressure, right? It starts from minus 60 to 70, goes to minus 40, right? And this movement of minus 60 to minus 40 is unstable, right? So it basically means that it slowly drifts towards the threshold, right? So it doesn't really stay at minus 60, it keeps going towards minimum so that it can depolarize, right? And why does this? So any depolarization is because of ionic movement. So after your repolarization, when your potassium has been moved out, things should come back to normalcy until the cell receives the impulse, there is a depolarization at the membrane receptor or membrane level, then your sodium channels should open and then sodium influx should take place. That is how depolarization takes place. But the SA node doesn't wait for the signal. It polarizes, comes to minus 60 and then goes to minus 40. How did this ionic change happen? How did this shift in your threshold potential take place? That's because of three kind of mechanism. First is something called funny current. Okay. Now, what is this funny current? It's basically the pacemaker cells, which is basically the SA nodes. They have a special channel called HCN channel. Okay. These are called funny channels. Now, what do these HCN channels do? They allow sodium influx, okay, at hyperpolarized potential. So at minus 60, these channels start functioning. Originally, it should have been later when at minus 40 or at the reception of any impulse or any receptor membrane deep function, like activation. They should have undergone changes. But at minus 60, these special channels, they start to act funny and then they start to, like take in the Na plus, where your depolarization starts. So as your sodium is now coming inside cell, the cell starts depolarizing and it is already at minus 60. The more Na plus comes inside, it starts depolarizing more, right? So it reaches minus 40. That's one. Second is you have a calcium channel, which is of two type, L type and T type, right? So your T type channels, they open near threshold and accelerate depotential. And the L-type channels, they open at threshold and then generate the upstroke of the action potential. So they are also contributing towards this action potential. So that's calcium channel. You have an HCl channel. They also have calcium channels. And also they have reduced potassium conductance. So basically they have a lower outward K plus compared to other ventricular cells. Because they have a lower K outward K plus, they have slow repolarization. So because they can't repolarize, they do more of depolarization, and the funny current contributes to the to it mostly. So overall, you have these funny current, calcium influx, and the low potassium outflux that together contribute to this unstable resting membrane potential, and that keeps going negative and negative. And because it goes negative, it reaches the threshold quickly, and it can by itself start generating impulse. Now, why is this different from the ventricular myocytes? That's because ventricular myocytes have a stable resting membrane potential. It is minus 90, it stays minus 90, right? Where is the ventricular myocyte? These are the ventricles contraction. So remember in the cycle, how does ventricles contract? When the stimulation has come from the Purkinje fibers. The Purkinje fibers will send an impulse and the ventricles muscles will start contracting. So they require external stimulation because they have a very stable resting membrane potential that will not keep fluctuating on its own. It needs an external stimulation. The other SA nodes, pacemaker cells, don't need that. So that's the difference. And that's why heart is a natural pacemaker. Okay. Right. So now that I've explained to you, this spontaneous depolarization of pacemaker cells would be primarily due to funny current, which is via sodium, we've discussed, and calcium leak channels. Leak channels are basically means that they are continuously moving in and out, right? Okay. Now, the other options that are given here, which is like fast sodium channels, which are basically, if there are fast sodium channels, they basically are inactivating the pacemaker cells, right? So these are not present in the pacemaker cells because they would inactivate the sodium channels, right? And you don't want that. Next is potassium influx. Efflux is for depolarization. It cannot be for depolarization. And ATP is activity, which is basically telling that it maintains the ion gradients, but ion just maintaining the ion gradient doesn't necessarily means the depolarization of the cells, right? Okay. Moving on to the next question. Okay. Which of the following best explains why calcium influx in pacemaker cells is essential? Why do you need? We have been talking about calcium influx. We just now said, right, leaky calcium influx. So why do you need that? I explained that to you, but let's just do it once more so that it becomes more clear. So the answer is it contributes to the upstroke. Upstroke is basically the beginning of the QRS, if you remember, right? So what does it say? It's basically, it says that the pacemaker cells, so in these cells, the upstroke, which is the phase 0 or the QRS early stage, uh, is due to calcium influx, right? And this is via the L-type channel that we discussed. And what do they do? Remember, it's depolarization. So what do we have to do? You basically contract. So calcium ions trigger contraction in the atrial myocardium downstream. So it contracts the atrium, pushes it down in the ventricle. And then because it pushes in the ventricle, the next QRS phase starts, and which further leads to the ventricular contraction, right? Okay. Now, the other option which says calcium influx prevent hyperpolarization, that's incorrect because potassium efflux repolarizations, right? The calcium doesn't have anything to do with hyperpolarization, or it does not prevent hyperpolarization. It basically means it does not do anything for that. Next says it maintains potassium balance. That's for your like it's for the sodium potassium pump. I know what you will be thinking. You will be thinking that we said somewhere that it is balancing out, but that's at the plateau stage. We are talking about pacemaker cells, right? So we are talking about why do you need it first of all, right? There we were talking about what happens in pacemaker cells. In there also it is doing the same thing. It's still contracting, but it is like it is at the beginning. It is delaying the efflux, right? So it is not striking against potassium balance. It is just till your potassium efflux is taking place, it is continuing to contract, right? Lastly, it says it opens sodium channels in atrial myocytes. So, well, just calcium influx doesn't do that. Calcium influx is only for your contraction. But there are still differences. So you need to remember it distinctly. Okay, next one. The action potential in ventricular myocytes. Keep remembering ventricular myocytes as the ventricular contraction. So the action potential here is different from the SA node. Why? So the ventricular myocytes depend on sodium influx for phase 0 depolarization. What does that mean? It means that it needs external stimulation, right? That's what we discussed. Okay, next. Okay, now this is about your ECG. So there are 4 questions on this slide. I will read out the question, wait for few time, and just let you imagine each of these different waves in your ECG and then try to correlate what does that wave represent. Okay, so first one is P wave. What does that represent? QRS complex, what does that refer to? T wave, what does that refer to? And the PR interval, what does that refer to? Right. So I hope that was clear. Let me give you the answer here first. Okay, so the ECG P wave refers to atrial depolarization, right? That begins, that is the first wave that we see. Okay, and then you have the entire QRS, right? So the next one, QRS, This one refers to the ventricular depolarization, right? T wave refers to the ventricular repolarization, if you remember, right? And then the PR interval is basically the difference between atrial depolarization and ventricular depolarization. Okay, right? All right, the next one says a monophasic action potential recording from a ventricular muscle fiber typically shows. Monophasic means one this thing, phase. Okay, let me give you the answer and then explain. So a monophasic action potential from ventricular muscle fiber shows a plateau phase due to calcium 2 plus influx. What does it mean? It basically means that ventricular muscle fibers, uh, it basically exhibit a prolonged plateau phase that we know of, and it is because of sustained calcium influx, right? But the key here is that it is from a ventricular muscle fiber, not from a pacemaker. In pacemaker cell, you will not see this feature of a plateau phase, right? And this prolonged plateau phase is crucial for the contraction of ventricles, right? Which is to allow efficient injection of blood. We have been already discussing this. This is just in different, in other terms, saying that at every step, be it atrial ejection or ventricular ejection, the step is regulated enough so that it ensures that the particular chamber is completely empty before moving to the next phase, right? Okay. Now, so far we have been discussing about ECG, ECG, ECG. So what does ECG actually do? Why is it actually used? So it is basically to detect the electrical activity, and it diagnoses arrhythmias or ischemia, right? So it's basically because it is recording the electrical activity of the heart, it will provide all the associated information like heart rate, the rhythm, the conduction abnormality, or any such changes, right? But it has to do with the electrical activity. It does not do any direct measurement of contraction or blood pressure or intracellular iron level. It just looks at the electrical activity, and based on the irregularities in the graphs, you can you can suspect that this would be a cause or irregularity in your heart. Then further checkups are done. But ECG basically gives you an idea if your all the chambers of the heart are functioning at a proper scale or proper level. Yeah. Okay. The next is, which of the following differentiates pacemaker action potential from ventricular action potential? In another term, same question, repackaged in different ways. And by now we already know that pacemaker cells show automaticity due to unstable resting potential. Automaticity means it is able to spontaneously depolarize. Spontaneously depolarize means automatically depolarize, so automaticity, right? So your ventricular myocytes, on the other hand, as we know, require stimuli to depolarize because they have a stable resting potential membrane, membrane potential, right? And pacemaker cells, as we know, they mainly rely on calcium influx for depolarizations. But that happens because they already have those channels which can function at their minus 60 millivolts, right? Okay. Next, which component of the conduction system directly stimulates ventricular conduction? So in case of all these different components, which one is the one that directly acts on the ventricular conduction, which has to be towards the end? Purkinje fibers. Okay. Aortic valve, right? I told you arteries don't have valves except for the two arteries that take blood away from the heart, right? So one of them is aorta. So what does valve do in general? Wherever you see valve, it has the same function. Prevent backflow from the aorta to the left ventricle. Okay. Okay. So the refractory period in ventricular myocytes prevents what? So what is the refractory period doing? So refractory period. So this is a connection to what we've already studied a few classes before, right? Tetanic contraction. So try to tetanic contractions or your atrial contractions, calcium influx. All of these are topics that we have discussed in several classes. So can you make a connection and try to think why this is important? We have only a few slides remaining. So maybe I can wait for a few minutes here. Let me again re-edit the question. So there's a refractory period in ventricular myocyte, right? What does it prevent? So the options are calcium influx, backflow of blood, atrial contraction, or tetanic contractions. Which one do you think makes sense here? So again, ventricular myocytes are basically the ventricles. So it's talking about the contractions and then followed by a refractory period. So why do you think it is important? Or in other terms, what do you think that it prevents? Okay, let me give the answer. So it's tetanic contractions, right? Now, what does it mean? So like I said, there's a refractory period in the ventricular myocytes. Where is this refractory period, the gap? It is a plateau phase before your T wave, which is repolarization, right? So what does it do? It ensures that the cardiac muscle, which is your heart, cannot be re-excited too quickly, thus preventing tetany and so it doesn't like it doesn't keep this twitching happening. It allows everything to fill and pump properly and for the next cycle to begin. Right? The refract, and it's important, therefore, that this refractory period happens because it is important for your coordinated contraction. Okay. Okay, so we begin the last set of questions and we should be able to finish soon. Right. So these questions are basically assertions and reasons. So you have two statements. One statement is assertion, which is basically a statement. The second statement tries to explain the first statement. And the question then follows if what is the relation between the two statements. So for here, you have first statement saying left ventricle walls are thicker than right ventricle walls. And that's because left ventricle wall pump blood to the entire body at high pressure. So do you think this is a correct statement individually, and are they explaining each other as well? Yes. So the answer is yes. That's what we have been discussing. So for your left ventricle has to pump out the blood to the uh, arteries that take the blood to the entire body. So of course it has to be at higher pressure, and because it is at higher pressure, the walls have to be thicker and elastic. Next one, pulmonary veins carry deoxygenated blood because pulmonary veins bring blood from lungs to the left atrium. So I hope you know the answer. It was A is false and R is true. So the pulmonary veins carry deoxygenated blood. Is it correct? So A is false because pulmonary veins carry oxygenated blood, not the deoxygenated blood. Yeah. And the second statement, however, is true that pulmonary veins, they carry blood from the lungs to the left atrium, right? So they carry this oxygen-rich, oxygenated blood from the lungs into the left atrium. Okay. Next, the AV node directly stimulates ventricular contraction. The AV node only delays conduction. Purkinje fibers trigger ventricular contraction. So you can see one of these statements has to be correct. So which one is correct and which one is incorrect? We have been discussing so far that your AV node delays the conduction, further passes to bundle of hairs. From there, it goes to Purkinje fibers, which then triggers the ventricular contraction. So therefore, R is correct, but A is false. Okay, and the last question for today's session. The plateau phase in ventricular myocyte prolong action potential duration. This ensures sufficient calcium entry for excitation contraction coupling. Correct, right? Both of these statements are correct and both of these explain each other. In a sense, your plateau phase is there because you need your calcium entry, which is required for proper contraction, right? Okay. So that brings us to an end of this problem solving session. This is problem solving session 10. So we have two more sessions, which we'll be discussing week 11 and week 12. And we have one extra session for 2 hours where I will be putting out a summary of the entire 12-week course that we are currently discussing. And what I would do is I will just briefly give you the overview of how does this course flow through, what the main idea is, and not go over at all in the details of all these different, because the individual problem solving sessions do that. So I don't want to again give you the same information. I will just give you a link of how this flows, and probably we can look into some different kind of questions, but I think it's 2 hours, so the most part of the problem solving session will be covered by the mind maps, which will give you a flow between these different lectures and the topics. And then I can see if I can put in some interesting way of showing you the different topics. So yeah, with all the link, I will also try to highlight what each of these topics would mean. And this would be on 11th of October, before the last class, the 12th week class, right? So I don't want to take off the regular week for that, regular week classes for that extra class. The extra class will be on a separate day. So if you can, please do join, else the video will be available for your perusal. Okay, so yeah, we can end the class today here. Thank you so much for attending.